Nivedita Joshi is here by taking a video on the topic the search for genetic material. This is a topic from the lesson number 6 from class 12th biology, and there were a series of experiments to find it out what is the genetic material. You know it very well. There are two nucleic acids, DNA and RNA. Okay, later on this was found that the DNA is the genetic material. But initially, what kind of experiments have been done to prove this, we will go through. So in this video, you will get transforming principle, which is one of the proof for the DNA as genetic material. And this experiment, transforming principle or the experiment, it was done by the Frederick Griffith. So he did a series of experiment, and he used bacterium Streptococcus pneumoniae. The bacterium uh, was causing the pneumonia disease. So he selected this bacterium, and the results were miraculous one, and he got the transformation in the bacteria. So for the experiment he used two important things. One is mice and second is the bacterium Streptococcus pneumoniae. Streptococcus pneumoniae has two strains. One is S strain and other is the R strain. S strain, it was virulent. Virulent means it will cause the disease. That's why it is virulent. Means it is pathogenic. We can say in other words it is pathogenic. So S is pathogenic. And R strain is smooth. Why it is smooth? Because it is having a, capsule like cover on it. So that's why it is that's why it is smooth. And about the R strain, R means it is used for rough, because it is not having any kind of capsule on it. That's why it is called as the rough strain or R strain. So it was not pathogenic one, so non-virulent, and this was not causing any kind of pneumonia. Okay, so this was the this way they were classified. S means smooth, that is pathogenic, and R is rough, which is non-pathogenic. Okay, so he used both the strains of bacteria. Now what he did in the experiment? In very short, the experiment is described in this slide in very short, and it can be kept in mind by using few equations or few statements. So what he has done, first of all, he injected S strain bacterium in the mice, and he found that the mice died. And when he injected R strain bacterium into the mice, he found the mice were alive. Okay, mice were alive, means they didn't die. Then what he did, he killed the S strain bacterium by providing very high temperature or by heating them, and after killing he injected the S strain bacteria, means killed S strain bacterium into the mice, and he found that the mice alive. Later on what he did, heat killed S strain were mixed with the R strain alive bacterium. Okay, S strain killed one, mixed with the R strain alive, and then the mixture is injected into the mice body. he observed that the mice die. So at the end he concluded that some transforming principle is there, and which transferred, according to which the heat killed S strain, uh, means transformed the R strain, means there was something in the S strain bacterium which transformed the R1, in easy word, and as a result R strain bacterium were able to synthesize that capsule over it, and they converted R strain converted into the virulent bacterium, that is into the pathogenic one. Means something, in short, something was there in the S strain heat killed bacterium which has converted the R bacterium into the S bacterium. Okay, this he concluded, and that's why he has given the name transformation, because R transformed into the S1. Yes, of course, the after the experiment, proof was given to this Griffith's experiment, and the proof was given by the Oswald, Avery, MacLeod and McCarthy, and they worked to determine the biochemical nature of transforming principle. Means what was that in the Griffith's experiment. So in order to find it out, they purified biochemicals, all the biochemicals present over there, like proteins, DNA, RNA, etc., from the heat killed S strain bacterium, and they have done it using suitable enzymes. And after that, they discovered that digestion of protein and RNA didn't affect transformation, means there was no change, okay, no influence. So this was proved that the protein and RNA couldn't be the transforming substances, as these substances have not affected the process of transformation. Secondly, they find it out digestion of DNA with the DNase is an enzyme, but the digestion of DNA, it was done with the help of the DNase. So they find it that uh, DNA caused the transformation of R cells to S cells, that is DNA was the transforming substances. To this conclusion they reached when they find the digestion of DNA inhibited transformation. Okay, this was the conclusion. This was the conclusion. Means this was the observation. We can say कि when digestion of DNA it was done, they found that the process of transformation it was inhibited, and they concluded that the DNA is the hereditary material. Means this was the DNA only which which has transformed the R cells to the S cells. So this was just uh, discovered. So this was one of the experiment to prove that the DNA is the genetic material.